Okay guys, today we're going to continue looking at how we make cells. And we're going to primarily spend our time now talking about meiosis, however you want to pronounce it. And you've seen this before. This is the basic way that we're going to produce male and female sex cells, right? Gametes. You've heard that word before, I know. Okay? And one of the things is that this is very common for most living organisms, that we have sexual reproduction. However, there are some weird cases, such as some certain species of salamanders or mice or even blackberry bushes that do not have fathers. Their offspring don't have fathers. Now, how in the world do they do that? There's relatively rare instances where there's something called parthenogenesis. The mom will actually produce egg cells that are diploid, and they do this because there's no males in the environment. It's one of those issues where if you have an all-female group, you can't produce offspring unless you do this. Now, human beings can't do this, okay? Actually, the most complex organism that can do this is a lizard. You all heard of Komodo dragons, right? Komodo dragons rarely, but have been known to do this because there are no males in the particular environment where they're located. They're the most complicated organism I've ever heard of that can actually produce through parthenogenesis. Usually it's smaller critters like lizards, and again, there's some mice, and as well as several species of bush or plants can do that, excuse me. Okay? But it does occur. But again, they're all basically clones of the mother. Okay? There is some variation, obviously, because it can vary ever so slightly, some of it. But again, they're all going to be female. Okay? That's not going to work. So my question is, why? Why do we go through sexual reproduction? Well, it's actually very simple. To increase genetic variability. Think about your mother and your father at home. If you have brothers and sisters from the same parents, they don't look exactly like you. There are some differences between them. Again, you may look very similar. You may have a lot of similar characteristics because you both come from the same parents. But as far as you having identical characteristics, unless you're an identical twin, it's not going to happen. There's always going to create some variation, and that's a good thing. We want variability. It allows the species to thrive, flourish, and then when bad things happen to a species, somebody, because they are genetically a little bit different from somebody else, they can have the ability to survive. Plus, can you imagine if everybody looked the same? That would be kind of boring, wouldn't it? So there are some genetic variations, and that's the reason why we have sexual reproduction, to create that that genetic variability. Now, with meiosis, we're producing gametes. And the thing is, gametes is a word for sex cells, sperm or ova, right? And again, in plants versus animals, it's slightly different. But remember, the whole idea is to cut the chromosome number in half. So they only have one set of chromosomes. Remember that word we listed a couple of hours ago? The haploid and diploid? Those is what we're talking about, okay? Now, we're going to go through something called meiosis. Now, if you look at this animal life cycle, you start with an adult. They take cells that are diploid, and then they turn them into cells that are haploid, either an egg or sperm. Okay? And then we take those two cells, one from each parent, join them together to make a zygote, and that zygote will eventually develop into the adult. But they have two sets of chromosomes. But those sex cells, those gametes, have one set. Same thing with plants. A lot of them will create the actual plant that will be the sporophyte, and then it produces spores that go through and we convert from diploid to haploid. We produce gametophytes that turn into either sperm or egg. Now, when we think about plant and sperm, we think about pollen, right? There's stuff floating around in the air. That pollen is actually plant sperm. If you want to be gross about it, that's basically it. And then they form the zygote, and we start this whole process again. So basically, both plants and animals go through meiosis. They go through this divisional stage, okay? Now again, here is mitosis, right? We start with 46 chromosomes, but then we go through the S phase, the synthesis phase. We copy our DNA, right? Replicate it. So for a brief, and I do mean a brief little time, we have basically, since we got two sets, we got 92 chromosomes, okay? And then we split, go through PMAT, right? Prophase, metaphase, anaphase, telophase, and we split, and we have 2 cells that have 46. That's mitosis, right? So we start with an original parent cell that's 46. We end up with 2 daughter cells that also have 46. Okay? It's basically cloning, right? Pretty straightforward, pretty simple. Now, however, though, if you look at meiosis, it's a little different. We do the same thing. We duplicate the chromosomes. For a brief time, we have 92. Then we go through something called meiosis 1. We have PMAP, prophase 1, metaphase 1, anaphase 1, telophase 1, most of the time. And then we will actually split up into 46 chromosomes, chromatids. And then we're going to go through meiosis 2, prophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2. And then we end up with 1, 2, 3, 4 sets of cells that have 23. So we started with 46, one cell. We divided not once, but twice to make 4 cells that have 23, half the number of chromosomes. Okay? Whether it's to be sperm or egg. Okay? Pretty straightforward. Now, how does it do this? What's so great about it? It creates that genetic variability. One of the things I want to mention during prophase 1, remember we talked about homologous chromosomes, the chromosomes, one you got from mom, one you got from dad. They're called, like the number 1 chromosome from mom and the number 1 chromosome from dad came together, so now you have homologous chromosomes. And remember, they copy each other, so both of them look like that weird little egg-shaped structure. So I've got 2 of them. This is what I'm talking about, right? One from mom, one from dad, right here. Okay? But what they'll do is they'll get together and then they'll do something called swapping or crossing over. They'll actually swap little pieces of DNA. And if you notice here, by the time we get done, this guy's a little different from this guy. This creates genetic variability, okay? Now, then they line up. Remember in metaphase, well, in metaphase 1, those chromosome pairs line up, and then in anaphase, they split apart. So this gets 1 duplicated chromosome, this gets 1, this gets 1, this gets 1. Whoops, I'm sorry. And you create, all 4 of them now are slightly different. This red one has a little blue end on this side. This red one has a little blue end on this side. This blue one has a little red end on this edge. This one has so we get some differences. Now they're going to be different. They're not identical to what they were before. And this creates this creates variability. And these things are, again, which ones do you get? It's random. What does that mean? This one could come over here, this one could come over there. They could have been on either side. Which one does this cell get? It's literally by random. Which ones line up first where? When you remember when you were in elementary school, when you all lined up, you never lined up in the same order every time unless they specifically told you to. It's just wherever you get in line. Okay? This creates multiple possibilities of sperm and egg combinations. Okay? Multiples. And again, identical twins, no. Because again, they came from one fertilized egg that then divided and made 2 separate eggs. Okay? So genetically they're identical, so that's the only case where this doesn't occur. Okay? So, in males, we go through a process called spermatogenesis, the production, and the word spermato means sperm, and then genesis means creation, so the creation of sperm. You start off with something called a spermatogonium. You go through mitosis, produce something called a spermatocyte. The spermatocyte, and again it's still 2N, splits into secondary spermatocytes. Then we go through meiosis part 2 when we split, and we form structures called spermatids. They're not fully sperm yet, but they're getting close. You see they're all in. They have 1 set of chromosomes, and then they develop into the spermatozoa, which is the sperm cells that we think of. The ones with little tails when they let them move around. So that's spermatogenesis. The key difference, though, however, is with females. Because they go through something called oogenesis. Okay? There's this thing called an oogonium that goes through meiosis, or mitosis, produces a primary oocyte. It'll go through meiosis 1, a secondary oocyte. But I want you to notice, we got 1 secondary oocyte and another little cell called a polar body. Now, polar body is just, it's junk. Let's just be honest. It's not going to do anything. Then, that secondary oocyte goes through division. It makes an ootid, and another polar body. So we end up with 1, 2, 3 polar bodies that are not eggs. And then that oocyte develops into an ovum or an egg. So ladies, the difference between you and the guys is guys, every time they take one cell, will make 4 sperm. Ladies, when you take one egg, oligonium, pre-egg, you're going to only make 1 ovum. Okay? Which again is a good thing, because if you made 4 ova, every time you had a menstrual cycle, or every time you went through your ovulation, you'd ovulate, you lay out 4 eggs at a time. So think about this. You can get pregnant and have 4 kids every time. That's kind of rough. Humans, no. Doesn't work very well. We don't want 4 kids, do we? Not every time. Ooh, that would be rough physically on the female body. 1 child is difficult on the body at once anyway. Having 4 at one time would be very, very dangerous. Right? Now, however, unfortunately, there are things that occur called errors. Errors do occur in meiosis. Okay? One of the main errors is something called non-disjunction. Disjunction means to separate. Non-disjunction means they don't. I want you to look very closely right here. This is a karyotype of a person. You'll notice they got two number 1s, 2 number 2s, and so forth. But then you get right over here to number 21. What do you notice? 1, 2, 3. You call this Down syndrome. The actual scientific name is something called trisomy 21, because it's the 22nd or 21st set of chromosomes, and you got 3 of them. And we know what this leads to. This leads to this common condition that we see. Well, not super common, but pretty common. Everyone knows someone, or maybe have a brother or sister who has Down syndrome. And again, this is because instead of having 46 chromosomes, they got a third 21st chromosome. So they actually have 47 chromosomes. And this is what causes this particular disorder, right? Now, again, that's through non-disjunction. The chromosomes are supposed to separate during meiosis, but for some reason they don't separate, and instead of getting 1 from one parent, you get 2 from a parent, and then a third one from somebody else, or from the other parent, excuse me. So say you get 2 from mom and 1 from dad, you got 3 now. That's bad. This is what causes this. Now again, luckily, if you'll notice the size of the 21st chromosome pair, they're not very big. So this can actually occur. If it was on like this one right here, trisomy number 1, it probably wouldn't work. This person probably would never be born, because it's too much genetic information. Okay? Now, sometimes you can have extra or missing sex chromosomes. Now again, the last 2 set of chromosomes are what we call sex chromosomes, because they determine your biological sex, male or female. Okay? And you can have some too many, you can have some not enough. For example, if you are XXY, remember girls are XX, boys are XY. They have an X and a Y chromosome, girls have 2 X's. Well, sometimes because of non-disjunction, you can create something called XXY. It's called Klinefelter syndrome. What we have there is we have 47 chromosomes, but you'll notice right here, down at the the sex chromosomes, I got 2 X's and a Y. Now, what is this person? Genetically, they're XXY. They are still a male, okay? They still develop as a male, but the difference is they tend to be sterile, which means they can't have offspring, and they tend to exhibit more feminine characteristics. Their musculature doesn't grow as well. They may produce breast tissue. They just don't look very masculine, and I don't mean effeminate, but I mean those secondary sex characteristics, facial hair and a few other things, may not develop very well. Okay, and it's hard, and a lot of times it's hard to tell if somebody's like that. So a lot of people may have Klinefelter syndrome and not even know it. Okay? The opposite of that is Turner syndrome. They're XO. What that basically means is they don't have an XX, they don't have an XY, they just have an X. All right? They're missing either an X chromosome or a Y chromosome because it didn't get put in. That person is female. They have 45 chromosomes. They're female, but again, tend to be sterile, which means they can't have children. They tend to have non-feminine characteristics. They really just don't develop those secondary sex characteristics that make them very feminine looking. You see what I'm saying? And again, does this occur? Is it rare? It's pretty rare, but it does occur. Okay? It's not, oh my gosh, 1 in a billion people have it. There are probably hundreds of thousands, if not millions of people, probably hundreds of thousands in the United States that have it, and worldwide several million people are probably this way. I don't know the exact numbers, nobody, because a lot of times you don't know that you are this. A lot of people don't ever get tested for this. But it does occur. And you probably know somebody that is one of these or the other, and they may not even know that they are. Okay? But that's a non-disjunction when it comes to these sex chromosomes. Okay? Now another thing is, with chromosomal abnormalities, not whether or not you get the right number, but what happens to those chromosomes. One thing is something called a deletion. Say this is my original chromosome, A, B, C, and then you got the centromere D, E, F. Well, deletion, hey look, I'm missing something, aren't I? Missing the B part. Okay? Something's gone. Okay? Duplication, here we got the exact opposite problem. Instead of having 1 B here, we have 2. So we duplicated a section of the DNA of the chromosome, and now it's got 2 sets instead of 1 set on that little piece of chromosome. Then you can have something called inversion. Okay? Look at this. A, E, D, C, B, F. See how some of these got swapped around? They got swapped around. Instead of A, B, C, the C and the B and the E and the D got swapped around. And again, they literally inverted, flipped around. Okay? And then the last one is something called translocation. This is where we may move the gene and move it. Look at here. JKL. Look at this. This one's kind of all. Instead of having 3 and 3, we got 2 and 4. This may actually occur. And these are all problems that are associated with the chromosomes themselves, individual sections of the chromosome having problems. Not whether or not we get too many of the chromosomes or not enough of the chromosomes. Okay? But these types of things, deletion, duplication, inversion, and translocation, do occur. Most of the time, it may not cause a problem. You may never even know it, but there are a lot of diseases that can be associated with these problems with mutations in the chromosomes. Okay? So, again, this is meiosis. This is how we make sex cells, and then some of the problems associated with producing these sex cells. So again, most plants and most animals do it this way, okay? And it creates that genetic variability that we want with these plants and animals so that we're not all exactly the same. We're not basically clones of one another, okay? All right.